This is Absorption by Roots Part 2 for ICSE Class in Biology. So in this video I will be teaching you all the leftover topics, the ones which I left in the part 1 video. And see the first part, please check out that video. It explains all the basic concepts. So please check it out. What you will study in this video. Those topics which are not present in the screen are covered in part 1. Please check it out. So the first topic we will be covering in this video is called turgidity. Turgidity is as important as osmosis and you should study it thoroughly. Define turgidity. Turgidity is defined as the condition of a cell where it cannot absorb any more water, and in this condition its cell wall is completely distended, meaning stretched. Explain turgidity. When a root cell continuously absorbs water from the ground, it cannot absorb any more water at one stage. This stage is called turgidity, and when the cell is reaching turgidity, the cell is said to be turgid. For a turgid cell, you can imagine a balloon. When you fill balloon with tap water, there is one stage where you cannot fill anymore. It looks like it's going to burst out. So this stage is called turgid. Two more things to discuss about turgidity. One is an example of turgidity. The main reason why fruits and vegetables sometimes blast out and their seeds are dispersed, the main reason for this is turgidity. The pressure inside the fruits and vegetables sort of build up and they blast. So this is the example for turgidity. And the second thing is the state of turgidity. When a cell is turgid, there is some sort of equilibrium or a balanced state of the cell. That is, no water enters the cell nor leaves the cell. So it's sort of in an equilibrium state when the cell is in a turgidity state. Next topic is turgor pressure. Turgor pressure is the pressure inside the cell caused by turgidity. Very simple. But when this question comes in the exam, you don't define it like this. Turgor pressure is defined as the pressure of the cell contents on the cell wall. So this is how you define it. This turgor pressure is very, very important, as you will see in the future. It is the cause of it builds up root pressure, which is a very important, again another important topic in this video. Wall pressure. So if you remember, turgor pressure is defined as the pressure of the cell contents on the cell wall, but wall pressure is the pressure of the cell wall on the cell contents. So turgor pressure is the pressure applied by the cell contents, while the wall pressure is the pressure applied by the wall. Fourth topic in this video, and one of the most important topics, is called plasmolysis. Plasmolysis is defined as the process of contraction of cytoplasm from the cell wall caused due to loss of water when placed in a hypertonic solution. So contraction means shrinking, opposite of swelling. So fifth topic of our video is called flaccidity, and flaccidity is defined as the condition of a cell that is shrunken and is no more turgid. Such a cell is said to be flaccid. And the next topic, the sixth topic, is called deplasmolysis. Deplasmolysis, it is defined as the reversal or the recovery of plasmolysis, basically the opposite of plasmolysis. Place a cell in a hypotonic solution, endosmosis occurs and the cell swells up, and when it cannot absorb any more water by osmosis, it reaches the stage of turgidity. So when you place a cell in a hypotonic solution, the cell becomes turgid, while if you place the cell in a hypertonic solution, the cell shrinks, and due to plasmolysis it becomes flaccid. Topic of our video is daily practices of plasmolysis. In your book, the four practices are given, and the first one is salting meat, and the second one is adding salt to pickles. Both these examples serve for the main purpose, killing bacteria. When you add salt, salt acts like a hypertonic solution and it withdraws water from bacterial cells present in the meat or pickle, thereby killing it. So that's why pickles come for a long run and they don't get spoiled even after 2 months or so. So because there are no bacterial cells present in the pickle, because the salt absorbs the water from the bacteria cell and it doesn't allow it to exist. It doesn't allow it to survive in the pickle or the meat. So yes, these are two most common practices of plasmolysis. Third example is removing weeds. You would have seen in a playground someone will sprinkle the salt and that's to kill the weeds present in the playground. So when you add salt to the weeds, salt again acts like a hypertonic solution, and so it will be concentrated on the outside. So from the plant, water will come out and thereby killing the weed. So this is the third example, killing weeds. So again, salt's used here to absorb water from the plant, and so it kills the weed. So the fourth example is adding excessive fertilizers to plants. So what happens when you add a lot of fertilizers to plants? People think if you add a lot of fertilizers, it's just a lot of good to the plant, but that's not true. If you add a lot of fertilizers to the plant, that the excessive fertilizers act as a hypertonic solution again, and they absorb water from the plant, just like you add salt to the plant in the previous example. So just the same thing. So again, the water from the plants come out and they die. So this is the fourth example. Next topic in our video is uses of turgidity. So there are five uses of turgidity. The first one is turgidity provides rigidity. That's obvious, because when a cell is in turgid state, it will be fully expanded and it will be very rigid, very hard, because there will be pressure from inside going in an outward direction, thereby making it rigid. So that's obvious. The first one is turgidity provides rigidity, and the second one is turgor pressure helps the seed to push the ground and come up. So turgor pressure helps the seeds to push through the ground. It helps the roots to push through the ground. So yeah, that's the second one. And the third one is, like I said before, turgor pressure helps to build the root pressure. And the fourth one is turgor pressure controls the opening and closing of stomata. So I will explain it in detail with a diagram. Fifth and last use of turgor is very important. It's called loss of turgor. The example for this is Mimosa pudica. Mimosa pudica is the scientific name of a plant, and what is its significance in this lesson? When you touch the leaf of a Mimosa pudica for 4 seconds maximum, the leaf will immediately droop. So why is this? That's what we will be studying now. When you touch the leaf for 2 seconds, there will be a loss of turgor at the base of the leaf and the base of the petiole, which is called pulvinus. The base of the petiole is called pulvinus. Okay, so when you touch the leaf of Mimosa pudica for 2 seconds, the leaf will immediately droop, because due to the touch stimuli there will be a loss of turgor at the base of the leaf and the base of the petiole, which is called pulvinus. So that is why the leaf droops immediately. in the case of Mimosa pudica. So the Mimosa pudica example was just one example for loss of turgor. There are two more. Loss of turgor also explain why a flower turns towards the direction of sunlight, and also the loss of turgor also explains the mechanism of insectivore plants that trap their prey. Topic of our video is called root pressure. Root pressure is defined as the pressure built due to continued inward diffusion of water by cell to cell osmosis, which helps in the ascent of cell sap. This might seem to be a long definition, but I didn't memorize it. I just understand the concept, and I can easily say it. So what is root pressure? Root pressure is the pressure that is developed in the roots, obviously, and this pressure helps you to push the cell sap from the roots to the above parts of the plant. Like when you press the toothpaste when you press the toothpaste bottle to bring out the toothpaste, just like that, there's a root pressure acting on the roots to help the ascent of cell sap. So that's all. And how is this pressure built? It is built by osmosis. The cell to cell osmosis, the continuous cell to cell osmosis in the plants help to build this pressure. The cell to cell osmosis creates a condition of turgidity, and turgidity, as we saw, helps build root pressure, and so root pressure is caused due to cell to cell osmosis. That's it. You don't have to memorize or mug up anything. You just have to understand the concept, and you'll just automatically get it. Next topic is guttation. Guttation is a very, very favorite question in terms of examination. There are many possibilities where a question on guttation might come in your exam, so you should pay a lot of attention to guttation. So guttation is defined as the loss of excessive water due to root pressure that are found in the form of water droplets in the leaf margin. So root pressure is developed in the roots. So due to excessive root pressure, a lot of water goes straight up, and in the form of transpiration, they just go till the leaf veins, the leaf margin, and you can see the droplets. So this guttation occurs more commonly in early mornings. So you would have seen in many sceneries in the early morning, the sun rises, and the leaves will be wet even if no one waters it. So yeah, that's because of guttation. Guttation is caused by, so there might be a question in your exam, why are the leaves wet in the morning even though no one watered it? So the answer you should write is guttation. So it's very simple. The next topic of our video is called experiments. So before starting with the experiments, it's better if you know that each experiment has two setups: the experimental setup and the control setup. There may be n number of experimental setups, but there is just one control setup. So the experimental setup proves your statement, and the control setup controls the statement. That means it limits the statement. Okay, so yeah, there are 4 easy yet important experiments, and okay, yeah, let's start. The first experiment is to show that root is required for absorption. So that is very, very simple. To prove it, all you need is just a test tube with the water filled to some level, and a plant with its roots submerged in the water and add oil to prevent evaporation, to show that water does not evaporate. And water okay, yeah, okay. So yeah, this is the experimental setup. In the control setup, you need a test tube with water up to the same level and oil to prevent evaporation. In the experimental setup, after a day or two, the water level would have come down, while in the control setup, the water level would remain the same. So in the experimental setup, the water level would have come down because the root absorbs water and the water level comes down. But in the control setup, there is no plant to absorb water, so the level remains the same. So let's move on to the second one. So the second experiment is to prove that water is conducted upwards through the xylem. It is very easy. All you have to do is take a beaker filled with Adhesion solution, which is red color. So after that, you need to take a plant and submerge its roots in the Adhesion solution. So after a day or two, if you use a microscope to look at its xylem vessels, they would be stained red, and hence proved. So the third experiment is to prove that the conduction of water is through the xylem. So how do you prove it? Easy. Take two setups, setup A and setup B. Setup A is a potted plant without phloem but with xylem. But the second setup B is a potted plant with the same plant but with the phloem and without the xylem. So after some time, you will see that setup A having xylem will show no changes, but setup B will have its leaves drooped. Setup A will have its leaf stiff because it has water, but setup B will have its leaf drooped like this because it wouldn't have water. So yeah, this is the experiment number 3. Experiment number 4, the last one. This one proves that food from the leaves is conducted downwards through the phloem. So how do you prove it? Very simple. Take a potted plant with its phloem cut around the middle. So this is how the plant will look like. The plant will have its phloem on the top and the phloem on the bottom, but the phloem in the middle will be missing. So what will happen? How do you know that? How do you prove it? How does this prove our statement? So the food from the leaves will be sent down through the phloem, and as it comes down in the middle, it won't have the phloem. So the food will sort of bulge in the middle, and yeah. So that's how it is proved. Finally, we have come to our last topic of the video. I know this video is pretty long, but yeah, okay, let's not waste any more time. The last topic of our video is called forces contributing to the ascent of sap. So before looking at the forces, you need to understand why this ascent of sap is so important. The cell sap is contained in the roots, and you would have seen in the previous part, the first part of the video, and it's made up of water and minerals. So yeah, that's what makes it more concentrated. So yeah, the cell sap is made of water and mineral nutrients. So these nutrients from the roots should be able to reach the top of the top part of the plant, that is the leaves, so that they can grow healthily. To see the 4 forces. The 4 forces are root pressure, capillary force, cohesion or transpirational pull, and adhesion. So now let's look at the first one, root pressure. So we already saw root pressure 10 minutes ago. Root pressure is the pressure built in the roots so that it pushes the cell sap above, like I gave the example of a toothpaste. So yeah, that causes the cell sap from the roots to rise above. The second force is called capillary force. The capillary force is created due to the narrow diameter of the xylem vessels. Uh, the water from the roots, the cell sap rises above to replace the water molecules lost by transpiration, and it rises due to the narrow diameter of the xylem vessels. The key point is the water rises from the cell sap rises from the roots to due to vacuum uh, to replace the lost water molecules by transpiration. Third force is called transpirational pull or cohesion. So let's first understand the meaning of cohesion. Cohesion is the force of attraction between two like substances, and in our case it's the force of attraction between water molecules. So how does this what role does this force of cohesion play in our case? In this context, the force of cohesion is responsible for the ascent of sap. So consider our case. As the water molecules are lost by transpiration, more molecules of water come to replace them. So imagine one water molecule coming to replace another molecule that is lost by transpiration. So one molecule of water is lost by transpiration in the leaves, and so one molecule from the roots go up to replace them. So this molecule brings up another molecule due to the force of attraction by cohesion. So that molecule is bound to this molecule. So two molecules go together, and this molecule attracts another molecule, and hence it goes up as a chain, and the cell sap is rising. So that's how cohesion plays its role. Fourth and the last force is called adhesion. Adhesion is the force of attraction between two unlike substances. In our case, it's the force of attraction between the water molecules and the surface cells, that is the cells of the xylem vessels. So how does this play its role in the ascent of sap? So with the help of the force of adhesion, the water molecules can easily stick to the surface of the xylem vessel, and hence easily go up against the force of gravity. So that's how the force of adhesion plays its role. Finally, it is time to conclude this video. Hope you found this video helpful. If you have any doubts or queries, leave a comment or send an email to this email ID. It would be lovely if you rated my video on a scale of 1 to 10. Please leave your rating in the comment section. Thank you for watching.